Imagine, if you will, the human body is a sprawling, biomechanical metropolis. It is a city that never sleeps, powered by a complex grid of energy. In a healthy city, the traffic flows smoothly. Fuel trucks, which we call glucose, arrive at the factories, which are our cells, and they deliver their cargo efficiently. The gates open, the fuel goes in, and the city hums with life. But in type 2 diabetes, this city is in a state of gridlock. The fuel trucks are piling up on the highways, clogging the streets. The factory gates are rusted shut. The city is starving for energy, even though it is drowning in fuel. For decades, our approach to fixing this traffic jam was somewhat singular. We tried to force the gates open with insulin, or we tried to stop the trucks from arriving. But what if there was a way to communicate with the traffic controllers directly? What if we could send a signal to the city's central command, the brain and the pancreas, to reorganize the entire flow of energy? For years, we had one way to do this. A signal called GLP-1. It was a good signal. It worked. But scientists began to wonder, is there another signal? A partner? A twin? Today, we are going to dive deep into a landmark scientific event that answered that question. We are going to look at a clinical trial that pitted the reigning champion of diabetes treatment against a new challenger, a molecule that mimics not one, but two natural hormones. This is the story of the SURPASS-2 trial. It is a story of chemistry, of biology, and of a 40-week journey that changed the way we think about metabolic disease. Hello and welcome to my channel Clinical Trials Explained. Before we open the dossier on this specific study, we must first understand the battlefield. To appreciate what the researchers did here, we have to understand the Incretin effect. When you eat a meal, your gut doesn't just sit there waiting for food to arrive. It is an active intelligence. As soon as nutrients hit your digestive tract, your gut releases hormones called incretins. These are chemical messengers. They travel through the blood to the pancreas and say, "Attention, please. Food is incoming. Prepare the insulin." This heads-up signal is crucial. In fact, if you injected glucose directly into your veins, your insulin response would be much weaker than if you ate the same amount of sugar by mouth. What's the difference? That is the incretin effect. Your gut prepares your body for the fuel. In type 2 diabetes, this effect is broken. The signal is weak. The pancreas doesn't get the memo, and blood sugar spikes. For the last decade, the gold standard in treating this has been a class of drugs called GLP-1 receptor agonists. GLP-1 stands for glucagon-like peptide 1. You can think of GLP-1 as a master switch. It does three amazing things. First, it tells the pancreas to boost insulin production when sugar is high. Second, it tells the liver to stop dumping extra sugar into the blood. And third, and this is critical, it travels to the brain and says, "We are full. Stop eating." This mechanism has been incredibly successful. But human biology is rarely controlled by a single switch. There is another incretin hormone, a sibling to GLP-1, known as GIP. That stands for glucose-dependent insulinotropic polypeptide. For a long time, GIP was the neglected sibling. Early research suggested it didn't work well in diabetics. But scientists at the pharmaceutical company Eli Lilly had a hypothesis. They believed that GIP wasn't useless, it just needed to work in tandem with GLP-1. They theorized that if you could stimulate both receptors at the same time, the GLP-1 receptor and the GIP receptor, you wouldn't just get a sum of the parts. You would get a synergy. A compounding effect. They created a molecule called Tirzepatide. It is a single molecule, but it wears two hats. It acts as a dual agonist. This brings us to the core inquiry of our script today. We know GLP-1 drugs work. We have a very famous one called Semaglutide, which you might know by its brand names. It is a powerful drug. The question the researchers posed was bold: if we take this new dual-acting molecule, Tirzepatide, and put it head-to-head against the champion, Semaglutide, what happens? Does the addition of GIP actually matter? Or is it just biological noise? To find the answer, they designed SURPASS-2. Let's walk through the architecture of this study. Clinical trials are the rigorous stress tests of modern medicine, and the design of SURPASS-2 was quite specific. This was a Phase 3 trial. Phase 3 is the final hurdle before a drug can be widely approved. It involves large numbers of people and rigorous monitoring. The study was open-label. Now, usually, you hear about double-blind studies, where neither the doctor nor the patient knows who is getting what. In this case, they used an open-label design because the injection devices for the two drugs were different. They couldn't easily hide which drug the patient was taking. However, to keep things fair, the final adjudication, the judgment of safety in major events, was done by a committee that was blinded to the data. The duration was set for 40 weeks. That is nearly 10 months. This is important because weight loss and blood sugar control take time to stabilize. A study of only 8 or 12 weeks would not have told the whole story. The researchers set out to recruit a specific group of people. They looked for adults with type 2 diabetes who were already taking metformin. Metformin is usually the first line of defense, the infantry of diabetes treatment. The participants in this study had to be taking a solid dose of metformin, at least 1,500 milligrams per day, and yet, their diabetes was still uncontrolled. Their HbA1c, which is a measure of average blood sugar over 3 months, had to be between 7% and 10.5%. For context, a healthy A1C is below 5.7%. So these were people whose systems were definitely struggling to manage glucose. They were also required to have a body mass index, or BMI, of at least 25, though as we will see, the average was much higher. In total, they enrolled 1,879 participants. These volunteers came from all over, the United States, Argentina, Australia, Brazil, Canada, Israel, Mexico, and the United Kingdom. This international spread is vital because it helps ensure the results apply to a broad range of humanity, not just one specific demographic. Once enrolled, these nearly 2,000 people were randomly assigned to one of 4 groups. This is where the head-to-head battle begins. Group 1 received semaglutide at a dose of 1 mg once weekly. At the time of this study, 1 mg was the standard high dose for diabetes control. Group 2 received the new challenger, Tirzepatide, at a low dose of 5 mg once weekly. Group 3 received Tirzepatide at a medium dose of 10 mg. And Group 4 received Tirzepatide at a high dose of 15 mg. The researchers weren't just looking for improvement. They were testing for non-inferiority and superiority. In plain English, they first wanted to prove the new drug wasn't worse than the old one. Once that was proven, they wanted to see if it was statistically better. Let's pause the science for a moment and consider the human element. Who were these people? The average participant was about 56 years old. They had been living with diabetes for an average of 8.6 years. This is a crucial detail. These weren't people who were diagnosed yesterday. They had been managing this condition for nearly a decade. Their bodies had been under the stress of high sugar for a long time. Their average body weight was 93.7 kilograms, which is about 206 pounds. Their average A1C was 8.28%. Remember, the goal is usually below 7%, or even below 6% if possible. So, these participants were walking around with blood sugar levels that were consistently damaging their blood vessels, their nerves, and their eyes. For the participants assigned to the Tirzepatide groups, the journey started slowly. You cannot just give someone a high dose of these hormones on day 1. The body needs to acclimatize. They started at a tiny dose of 2.5 milligrams. Every 4 weeks, the dose was stepped up by another 2.5 milligrams until they reached their assigned target. This titration period is like slowly turning up the heat in a sauna. If you blast it immediately, it's intolerable. If you raise it slowly, the body adapts. Over the course of 40 weeks, these participants checked in regularly. They had their blood drawn. They were weighed. They reported how they felt. And slowly, the data began to accumulate. Now, we arrive at the reveal. The 40 weeks are up. The data is unblind. What did the researchers find? The primary outcome measure was the change in HbA1c. Did the drugs lower blood sugar? The answer was a resounding yes for everyone, but the magnitude of the difference was striking. Let's look at the control group first. The people taking semaglutide 1 mg saw their A1C drop by an average of 1.86 percentage points. That is an excellent result. It confirms why semaglutide is a top-tier drug. Bringing someone from an 8.3% down to a 6.4% is life-changing. But now, look at the tirzepatide groups. The lowest dose of tirzepatide, 5 mg, reduced A1C by 2.01 percentage points. Already, the lowest dose of the new drug was outperforming the standard dose of the competitor. The medium dose, 10 mg, reduced A1C by 2.24 percentage points. And the high dose, 15 mg, reduced A1C by a massive 2.30 percentage points. Statistically, tirzepatide was declared superior to semaglutide at all three doses. But averages can sometimes hide the real triumph. Let's look at the targets. In diabetes care, the holy grail is to get a patient's A1C below 5.7%. That is the range considered normoglycemic. It effectively means that, by the numbers, the person has blood sugar levels indistinguishable from someone without diabetes. In the semaglutide group, 20% of participants reached this normoglycemic target. 1 in 5. That is respectable. In the high-dose tirzepatide group, that number was 51%. Let that sink in. More than half of the people who took the highest dose of this new medication saw their blood sugar levels normalize completely. They essentially achieved a biochemical remission of their high blood sugar during the trial. While blood sugar was the primary goal, the secondary goal, weight loss, is often what patients care about most. Type 2 diabetes and obesity are deeply linked. They are often two sides of the same coin. Breaking the cycle of obesity often breaks the cycle of diabetes. Here, the divergence between the single hormone and the dual hormone became even more apparent. Participants on semaglutide lost, on average, 5.7 kilograms. That's about 12.5 pounds. Again, a very solid result. But the tirzepatide groups were in a different league. The 5 mg group lost 7.6 kilograms. The 10 mg group lost 9.3 kilograms. And the 15 mg group lost 11.2 kilograms. That is nearly 25 pounds. To put this in perspective, the high-dose tirzepatide group lost roughly double the weight of the semaglutide group. When the researchers looked at how many people lost at least 15% of their body weight, a substantial amount that can reshape metabolic health, the numbers were stark. Only roughly 8% of the semaglutide group achieved this. In the high-dose tirzepatide group, nearly 40% achieved this level of weight loss. This data suggested that the Twincretin theory, the idea of adding GIP to GLP-1, was not just a theory anymore. It was a physiological reality. The two hormones were working together to burn fat and regulate appetite in a way that one hormone simply couldn't match. However, no medical intervention is without cost. We must discuss safety and tolerability. When you manipulate the digestive hormones, the side effects are almost always found in the gut. The human stomach is sensitive. When you slow down gastric emptying, which is one of the ways these drugs work, you can feel full, but you can also feel sick. In SURPASS-2, the safety profile was scrutinized heavily. The most common complaints across all groups were nausea, diarrhea, and vomiting. Nausea was reported by about 18% of the semaglutide group. In the tirzepatide groups, it ranged from 17% at the low dose to 22% at the high dose. So, the nausea rates were comparable, perhaps slightly higher for the new drug, but not drastically so. Diarrhea followed a similar pattern, affecting roughly 13 to 16% of participants across the board. Most of these side effects were classified as mild to moderate. They typically happened during that titration phase we talked about earlier, when the dose was being increased. For many people, these symptoms subsided over time as their bodies adjusted to the new hormonal reality. However, for some, it was too much. The study tracks discontinuation due to adverse events, essentially, how many people quit because they felt terrible. In the semaglutide group, roughly 4% of people quit due to side effects. In the high-dose tirzepatide group, that number was roughly 8.5%. This is an important data point. While the new drug was more effective, it was also slightly harder to tolerate for a subset of patients. It underscores a fundamental truth of medicine: there is no free lunch. Stronger potency can sometimes mean a harder adjustment period. The researchers also looked for more serious issues. They monitored the pancreas for inflammation, known as pancreatitis. This has been a theoretical concern with incretin drugs for years. In this trial, the cases of pancreatitis were very rare and evenly distributed across the groups, suggesting no elevated risk in this specific timeframe. They also watched for hypoglycemia, low blood sugar. This is the dangerous flip side of diabetes treatment. If you lower sugar too much, the patient can pass out. Remarkably, clinically significant hypoglycemia was rare in all groups. Because these drugs rely on glucose to work, remember, they are glucose dependent, they generally don't hammer blood sugar down unless it is already high. This is a massive safety advantage over older drugs like insulin or sulfonylureas. We have the data: tirzepatide outperformed semaglutide. But why? Why did adding GIP make such a difference? To understand this, we have to look at the fat cells themselves. We know what GLP-1 does. It stops appetite in the brain and slows the stomach. GIP is more mysterious. Historically, GIP was thought to actually promote fat storage. But that was in the context of a healthy person eating a high-fat diet. In the context of this drug molecule, and in the context of a diabetic body, GIP seems to do something different. The current scientific consensus suggests that GIP may act directly on the adipose tissue, the body fat. It may improve the sensitivity of fat cells to insulin, preventing them from breaking down and flooding the blood with fatty acids. It also seems to have a powerful synergistic effect on the brain's nausea centers. Some researchers believe that by stimulating the GIP receptor, you can actually tolerate a higher dose of GLP-1 agonism. It's as if GIP calms the nausea center just enough to allow the GLP-1 part of the molecule to do heavy lifting on weight loss. Furthermore, GIP is a potent stimulator of insulin secretion, even more so than GLP-1. By hitting the pancreas with both signals, the beta cells, the cells that make insulin, are engaged more robustly. It is a biochemical symphony. GLP-1 conducts the rhythm of the stomach and the appetite. GIP harmonizes with the fat cells and the pancreas. Together, they reset the metabolic thermostat. So, what does SURPASS-2 mean for the world? This trial was a pivotal moment in the shift from glucocentric treatment to weight-centric treatment. For 50 years, the goal of diabetes care was simply to lower the sugar numbers. Doctors would prescribe anything to get the A1C down, even if the drug caused weight gain, which insulin and some pills often do. SURPASS-2 solidified a new paradigm. It showed that we can treat type 2 diabetes by treating the underlying weight and metabolic dysfunction. By achieving double-digit weight loss, 10, 12, 13%, we aren't just masking the symptoms of diabetes. We are reversing the driver of the disease. The fact that over half the participants on the high dose reached normal blood sugar levels challenges the old notion that type 2 diabetes is a relentless, progressive disease that only gets worse. This data suggests that with the right hormonal intervention, the clock can be turned back. It also sets a new bar for competition. Semaglutide 1 mg was the standard. SURPASS-2 showed that the ceiling is much higher. This pushes other scientists and companies to innovate further. If 2 hormones are better than 1, what about 3? And indeed, triple agonists are currently being studied in labs right now. Now, as we approach the end of our analysis, we must be responsible. This script is for educational and informational purposes only. It is not medical advice. I am a narrator explaining a scientific publication, not your doctor. While the results of SURPASS-2 are exciting, every medication carries risks. Tirzepatide and semaglutide are powerful drugs with contraindications. For example, they are not recommended for people with a personal or family history of medullary thyroid carcinoma or multiple endocrine neoplasia syndrome type 2. Furthermore, these drugs are not magic wands. In the trial, the participants were also counseled on diet and lifestyle. The drug is a tool, an engine booster, but the patient is still the driver. If you have diabetes or concerns about your metabolic health, you must speak with a healthcare professional who understands your unique medical history. Do not change your medication based on a video or an article. The SURPASS-2 trial is already etched in the history books of endocrinology. It was the moment the Twincretin era truly began. It painted a picture of a future where type 2 diabetes is not managed through a daily struggle of finger pricks and failures, but through a weekly reset of the body's own hormonal intelligence. We saw nearly 2,000 people step into the unknown. They endured nausea. They endured injections. They devoted 40 weeks of their lives to science. And thanks to their participation, we now know that it is possible to achieve levels of control and weight loss that were once thought impossible without surgery. The city of the human body is complex. The traffic jams of diabetes are stubborn. But with trials like SURPASS-2, we are finding better ways to clear the roads, open the gates, and let the energy flow once again. Science moves forward, one data point at a time. And in this case, the data points spelled out a new hope for millions. Thank you for listening to this deep dive into the science of SURPASS-2. Stay curious, stay informed, and keep watching the horizon.